Hey y'all. We will be talking about experiment 21, catalase production right now. Um, and the purpose of this experiment is to determine which cells have enzyme catalase and why catalase is important to cells. And it's a very, very simple experiment. So kind of easing our way back into the second half of this semester. All right, so a little bit of background real quick is that so enzymes are what you call biological catalysts, and you kind of learned about them in 1201, and you might have talked about them in lecture class already. But the way that they work is that you have this red thing called a substrate enter in the active site of an enzyme right here. And once that substrate is there, the enzyme, we know biological catalysts lower activation energies. So here, once the substrate's in that enzyme, it's going to lower the activation energy from this black line way up here. It's going to lower it all the way down to where it's at this red line. So it takes a lot more energy to start this reaction to proceed to the products. So once the activation is lowered, the activation energy is lowered, the substrates will then go through the reaction and produce the products, and then the enzyme will release those products. And there's a whole bunch of different kinds of enzymes, but the two major categories that we talk about are extracellular and enzymes, and inducible and constitutive. So extracellular enzymes are ones that are excreted from the cell. So the cell will make the enzyme and then just excrete it to the outside environment, and then the enzyme will act on substrates on the outside of the cell. And there's endoenzymes that work more on the inside of cells to degrade molecules into like simpler products. Then there's inducible and constitutive. Inducible, we learned about, if you remember talking about the lac operon or any of the operons that we talked about how you have inducers and activators that help either turn on or turn off genes. So if the gene codes for these enzymes, then that enzyme is an inducible enzyme because it's controlled by an operator and an operon and all that. A constitutive enzyme is just always produced. There is no control mechanism. It's just constantly produced. Constitutive is a term that we say in biochemistry of something that is just always produced in a cell. You have like constitutive receptors, and in this case it's constitutive enzymes. So it's just always produced no matter what the conditions are. Inducible is controlled by the conditions of the outside environment. Now, just a review of metabolism is the sum of reactions that are associated with life, and it's definitely mediated by enzymes. The breakdown, and it's deals with the breakdown and buildup of macromolecules. Alright, so we talked about in 1201 and probably again in the lecture class already, we talked about metabolism, and we go over and we see this chart right here where you go in as glucose and then you undergo glycolysis and produce 2-pyruvate, and then you produce some ATP and NADH right here. We can use this as energy now. That pyruvate can then go into the mitochondria, and the mitochondria undergo pyruvate oxidation where you produce 2-acetyl-CoA, and then you'll produce 2-NADH, which are electron carriers that we produce the same here, and that we'll talk about that later. And then these acetyl-CoAs will then enter the citric acid cycle, and the citric acid cycle will then use up the rest of those carbons and produce them as carbon dioxide, and then we'll produce some more electron carriers as well as some ATP as well. Then these electron carriers that we've been talking about will then enter the oxidative phosphorylation and electron transport chain right here. Once they go into the electron transport chain, which we'll talk about more later, they will then be donate their electrons, and the electrons will move through the electron transport chain, creating a proton gradient, and that proton gradient will then power chemiosmosis, which will then diffuse hydrogens through ATP synthase, and then the ATP synthase will then make 32 or 34 ATP depending on what you're going to produce there on certain environments. But this is kind of like catabolism in a nutshell right here of oxidative respiration. And here we have glycolysis right here. So we have glucose going to pyruvate. These are the whole entire extensive process in all its glory. So here you can see glucose right here, and then it goes through all these steps, and then you get 2 molecules of pyruvate. This just gives you an idea of how extensive this process is. But each one of these enzymes right here, hexokinase, phosphoglucoisomerase, everything in blue is an enzyme that catalyzes a reaction. So like we saw in the last thing of the last slide right here of this substrate going to this enzyme and then releasing as these products, that step right here is this step right here, and then these products will then be the substrates of this enzyme right here to produce this product, and then this product will then be a substrate of this enzyme to produce this product, and so on and so forth, all the way down until you reach pyruvate. So it's a very elaborate process, but it's all mediated by enzymes, and each one of these processes, glycolysis, pyruvate oxidation, citric acid cycle, oxidative phosphorylation, and chemiosmosis, they're all catabolized by enzymes. So it just gives you the importance of enzymes as a whole. Alright, so that's just a little background on enzymes, but then we're going to start talking about physiology. So we talked about a lot the first half of this class. We talked about morphology a lot with the cell shape and arrangement. We talked about cellular morphology and colony morphology, and that's how a lot of biologists identify certain microbes. But that's only one kind of view of how we identify stuff. The other view is physiology, and it's talking about the metabolic capabilities. So we talked about glucose here, but then there's other different ways that microbes can survive outside of just metabolizing glucose. And so examination of these enzymes and microbes and the products of the reaction can give you a look into their physiology. And it's a lot of what we talk about in the second semester. So this experiment that we're going to talk about today is one of the physiology experiments. Alright, so let's just continue on aerobic respiration. So we talked about glycolysis, citric acid cycle, pyruvate oxidation, and this is the electron transport chain right here. So as these electron carriers are donating the electrons to this electron transport chain, the electrons move through the chain, producing a proton gradient. So protons get pumped across the membrane. But at the end of the electron transport chain, you have oxygen right here coming in and being the final electron acceptor of the electron transport chain. And that's how aerobic respiration works. And that's why we have to breathe as individuals, but that's also how microbes survive. And what happens is that these oxygens accept the electrons and then produce H2O. And then you can take this proton gradient, pump it across ATP synthase, and make ATP for energy. At the end of the electron transport chain, most of the oxygen accepts electrons, and they do form water. But sometimes, instead of forming water, they form hydrogen peroxide. Now, this is very bad because hydrogen peroxide is toxic to organisms. However, organisms do have a protective mechanism by producing the enzyme catalase. So enzyme catalase takes hydrogen peroxide, and then hydrogen peroxide enters the active site, lowers the activation energy, and produces H2O and O2. So it produces water and oxygen. Neither one of these are toxic to organisms, so it can detoxify this H2O2. And this happens in nearly everything that's aerobic respiration, that goes through aerobic respiration, produces catalase, nearly all of them. So, and then anaerobic organisms that don't undergo this do not produce catalase because they don't need to, because they don't have any H2O2, because they don't have this electron transport chain. So this enzyme catalase is very important in the role of life for anything that produces energy by aerobic respiration. So classifying which organisms have and which organisms do not have catalase is important in determining to give an indication of the way that they kind of harness energy. So what we did here in this experiment is we determined which microbes have catalase and which don't. So what we did was we put a big old drop of hydrogen peroxide. We took a slide, a glass slide that we used from microscopes, and we just divided it in half with a marker, and we put on both halves, we put hydrogen peroxide, like a big old drop of them. All right, and what we did was we took our inoculating loop and we took Staphylococcus mutans and added it to this hydrogen peroxide drop, and we took Staphylococcus epidermidis Oh, wait. This is a typo. This is supposed to be Streptococcus mutans and then Staphylococcus epidermidis. Sorry about that. So it's Streptococcus mutans and Staphylococcus epidermidis. So we took Streptococcus mutans and added it to this hydrogen peroxide and Staphylococcus epidermidis, and added it to this hydrogen peroxide. And what we found was that Streptococcus mutans just kind of laid there and nothing really happened. But with Staphylococcus epidermidis, we saw the production of a whole bunch of bubbles right here. And what these bubbles are oxygen that you can see from this reaction right here. So if you see the production of bubbles, you know that this reaction is occurring because you see the O2 bubbles form. Streptococcus mutans does not have any bubbles, so we know that it's probably an anaerobic respiration. Anaerobic respiration, and then these cells are probably just dead because they're exposed to the toxic hydrogen peroxide. But these cells might still be in a relatively living state. There's a lot of hydrogen peroxide, so it probably killed it. But you can see the results that this has O2 bubbles and this don't. So, just like that, the results are that Staphylococcus and Micrococcus are catalase positive and aerobic. So, our Staphylococcus epidermidis are aerobic, so they produce bubbles, so they have catalase. Streptococcus, Lactococcus, and Enterococcus are all catalase negative because they're primarily fermenters. They do fermentation to live. So, Streptococcus mutans is fermentive, and so it doesn't have catalase. So, the takeaway here is that aerobic respiration, organisms that undergo aerobic respiration produce toxic hydrogen peroxide as a result of that aerobic respiration. Catalase can then break down that hydrogen peroxide and detoxify it. So the way that you test for this is by mixing a culture with hydrogen peroxide and see if oxygen bubbles are produced. If there are oxygen bubbles, then we know that they have catalase. But if there are no oxygen bubbles, then we know that they're anaerobic respiration. And we'll be looking at a lot more of these physiology tests coming up.